Chemistry, we've got, first of all, metabolism. Good thing in biology to know stuff about. We know that our body is composed of cells. Many types of cells. And those cells are composed of many molecules. I was told by last hour that you haven't really talked about the differences between animal cells and plant cells yet in biology, but you'll get there. We're not going to focus on that too much. We're focused more on the chemical reactions that are going on, less about the cells that are within or the parts of the cells. And those chemical reactions are all revolving around those molecules, because often the molecules that are within the cells are interacting with each other. Makes sense. We've seen many macro examples of things that are interacting and causing reactions. It makes sense that if we put a bunch of molecules within a cell that they're probably going to interact. And when they're interacting, what they're generally doing is undergoing chemical reactions. And that's what we're focused on when we're talking about biochemistry, the chemistry aspect of it. So we know that we have cells. We know that the cells are composed of many molecules. We know that the molecules interact with chemical reactions. The sum of all of those reactions is known as metabolism. Another big word for us is called respiration. Give me a thumbs up if you've heard that word before, a thumbs down if you have no idea what it is. Awesome. Hopefully you've heard of it and know that maybe you know, that it's involving redox reactions. So thinking back to last year, topic 9 in IB Chem, we have redox, composed of two types of reactions that are always interconnected. Can't have one without the other. We have the reduction half of the reaction and the oxidation half of the reaction. And it's all involving the transfer of electrons. So thinking back to last year, the red stands for reduction. Is that involving losing or gaining electrons? Reduction. Gaining, absolutely. So we've got the rig part of oil rig. Okay, they're gaining electrons. So the oxidation is what? Loss, absolutely. Oxidation is loss. Reduction is gain. Redox is oil rig. Way to think of it. So we have the transfer of electrons. One thing is giving up their electrons to something else, and so I have a net change in the oxidation numbers of one of my substances and my other substance as well. All of these respiration reactions are involving the release of energy, so they are all exothermic. So if we think back to chapter 5 or topic 5 from IB, we're dealing with heat, energy, all of those things. And I have my delta H values. I have my little energy level diagram. My delta H value, would it be positive or negative? Negative, very nice. So exothermic, my delta H value is negative. So my change in my energy is negative. Okay, we know in respiration that carbohydrates are converted to glucose. And we'll talk more about sugars and carbohydrates later. And that glucose is then used by the cell for those reactions, those redox reactions that are exothermic. Oops. We can test what's going on with different food sources using calorimetry. And we did that last year, not using food sources, but we did it figuring out the specific heat of copper. We did that lab last year. Many of you that took APES did some calorimetry where you combusted a substance within a bomb calorimeter and you were looking to see how much energy was released. At least that's what I've heard from the APES teachers. So we know that humans need energy for their cells to function properly. So it can be very important to know how much food is stored within a food item so that we can make sure that we're getting enough calories in our daily diet. I know I need those calories, or I need that energy. I need those sources of energy. So I'm going to make sure that I get enough of that energy into my system. When they figure that out, we don't know specific heat capacities of different food sources. We don't know those things, and that's not important for us because we can figure out the energy based off of a change in water. And that's part of what you were seeing last week when you were working on that Q is equal to MC delta T worksheet, depending on which problem you were working on. So we do all of this in a bomb calorimeter. So I take whatever my substance is and I burn it. And as I'm burning it, it is transferring its energy to water. The water is what I know a bunch of stuff about. I know, or I can measure, the change in the temperature. I know the specific heat capacity of water. Who remembers what that is? Yeah, 4.184. Good. 4.184 joules per gram kelvins. So we know that about water. We would know what mass of water we have to start with. So we know all of this stuff about water, and it doesn't matter anymore that we don't know the specific heat capacity of whatever my other substance is, so what my food source is. The reason it doesn't matter is I can use this equation. So it's almost the same equation we had before. Q is equal to MC delta T. This time, though, we keep all of that MC delta T all for water, and we divide that by the mass of the food. And that figures out the heat transferred from that food. That would be a really handy equation to memorize. You might want to put a big star by it. And on the back of your page, we're going to do one example of it. Okay, here are the numbers for it. So we have a 0.50 gram sample of food combusted in a bomb calorimeter that raised the temperature of 100.00 grams of water 20 degrees Celsius. Ha, it's already given me my delta T. It didn't tell me T initial, T final. It's already given me my delta T, so I don't have to calculate change in temperature. It's right there for me, 20.0 degrees Celsius. And it's asking the energy of the food. So it's always good with these problems, start out identifying your givens. So my givens, I have the mass of water is 100.00 grams. My specific heat capacity of water is 4.184 joules per gram kelvins. My mass of food is 0.50 grams. And my delta T, oh, I forgot to carry through my significant figures. That should say 20.0 degrees Celsius. And since it's a change in temperature, taking final minus initial, my change is the same if it were in kelvins. So if I were to add 273 to both of them, the change is still going to be 20. It's just a numeric value. And since my C of H2O is in kelvins, I might as well just think of it as 20 kelvins rather than 20 degrees Celsius.
Wait. Oh, it doesn't matter
since it's just a change. Yep. Even if it didn't just give you a change and you calculated the change in Celsius, because it gave you an initial and a final, that would be the same difference that if it was in kelvins. Thank you for asking. Okay, so here's our equation again. Q is equal to the M of water times the C of water times the delta T of water divided by the mass of the food. There's no other units that we have to cancel here, which makes life a little bit easier. If you were to see this type of problem on the IB paper 2, the written section, make sure to show your work. That way if you have a mistake that gets carried through, you won't get that mistake carried through marked off. So make sure to show all of your work on your problems. Yeah, Jonah. Yeah, so since my temperature change would be just T final minus T initial, if I were to, let's say it was 50 degrees Celsius is my final temperature and 30 degrees Celsius was my initial. If I were to add 273 to each of those to make them both in kelvins and then subtract them like I would for my 50 and my 30, it's going to be a change of 20 no matter what, whether it's change in Celsius or change in kelvins. So since I'm dealing with change, Celsius and kelvins would be the same thing. Seen a lot of you type into your calculator already. This is your setup, your general setup if you put all of your numbers in. 100.00 grams times 4.184 joules per gram kelvins times 20.0 kelvins divided by 0.50 grams. Some of my units cancel out right away. I see that I have a grams on the top, grams on the bottom. Also kelvins on the top, kelvins on the bottom. So my unit's going to be a joules per gram. And I'm doing multiplication and division, which means with my sig fig rules, I'm looking for the lowest number of sig figs. So I want to see with your hands, I'm going to point to different numbers, I want to see with your hands how many sig figs are in that number. So looking at this first one, 100.00, show me with your hands how many sig figs are in there. You are right, all of those zeros are significant. There are 5 sig figs in that one. 4.184, show me with your hands how many sig figs are there. Good, they're all non-zero digits, so they're all significant. 4, perfect. And then we have 20.0. How many sig figs are in that one? Excellent. 3 is correct. The 20 and the 0.0 are all significant. And then we get to 0.50. How many significant digits do we have in that number? Good. I couldn't even throw you guys off. There are just 2 in there. So the 5 and the 0 are important. The 0 that's leading is not important. So that means my final answer has to have 2 sig figs. It's a pretty large number, so writing it in scientific notation is probably going to be the easiest way for you to represent it with just 2 sig figs. And hopefully